Hello, I'm going to talk to you today about the specific immune response, and in particular going to look at the cell signalling part of that. So last lesson we looked at the immune response, and we talked about the basic process, so I'll have a bit of a recap of that. And then we will look at the role of histamine again and the role of some new signaling molecules, so interleukins, monokines and interferons as part of the immune response. So last week we talked about the process of the specific immune response, which starts off with antigen presentation. So that's when a macrophage engulfs the pathogen and they will digest everything except for the antigens that it will then display on the surface. So it becomes this antigen presenting cell. That antigen presenting cell will then travel around the body until it comes across the correct type of B and T cell with receptors that are complementary to the antigen. So that process is called clonal selection. And the receptors will then bind to the antigen, and the cells then will become activated and they'll start to send signaling molecules. So today we'll be looking at those signaling molecules and their job. Those cells then that become activated will start to divide by mitosis. So they're making lots and lots of the correct type of B and T cells. And then they'll start to differentiate. They'll specialize into specific cell types. So at the top here, you can see we've got our antigen presenting cell, and through the process of clonal selection, then you get the selection of the correct type of B or T cell with a complementary receptor. Which will bind to the antigen. So this is often the T helper cell, but it doesn't actually have to be the T helper cell. There are other cells that could be involved in clonal selection process. That T helper cell then will activate other cells by sending signaling molecules and stimulate them so that they will start to divide by mitosis. Then you've got your clonal expansion process taking place, and those will then differentiate into different types of cells. So the B cells could become B plasma cells that produce antibodies, or B memory cells that will act as B immunological memory, T memory cells that act as immunological memory, or the T helper cells, or T regulatory cells, or the T killer cells. So those are the types of cells that the T cells could become. So B cells are called B cells because they will mature in the bone marrow, and T cells are called T cells because they mature in the thymus. So the T cells, the T lymphocytes then could become T helper cells. These are seen as the coordinators of the immune response. So they'll release these chemical messengers that we'll be looking at today, and they'll stimulate the B cells to differentiate. T killer cells then, they'll search for pathogens, they'll attach to the antigens on the surface of those pathogens, and then they'll produce toxins that will destroy those pathogens and infected cells. T memory cells then, they'll circulate in the blood for many years, so they'll act as immunological memory, which means if we get that infection again, then we'll be able to produce a much faster response. We call that the secondary response. So the T memory cells will help with that. And then you've got the T regulatory cells, which will shut down the immune response once the pathogen is destroyed and prevent autoimmune diseases. With the B cells, then you've got your B plasma cells. They'll develop into either produce they'll start to produce antibodies very, very quickly. So sometimes they're called effector cells, sometimes they're called B plasma cells, but plasma cells then produce antibodies very quickly. The B memory cells then are the other ones. So those again will circulate in the blood and act as immunological memory to produce a much quicker secondary response. In fact, it's so fast that those B memory cells can differentiate straight into B plasma cells and start to produce antibodies straight away. So much more antibodies produced much, much quicker than the primary response. So we're going to look today at the cell signalling, so the types of communication that you get between them. So this involves these chemical messengers that are sent between cells, and they'll travel to the target cells and they'll bind to complementary receptors and they'll stimulate a response. So there are lots of different molecules which have got different target cells and will bring about different responses. And one that we've looked at already is histamine. We know that the role of histamine is to cause leaky capillaries and attract neutrophils to the site of infection. So we're going to be looking at a group of chemicals that are called cytokines. These are hormone-like proteins that are produced by cells, and they're used for cell-to-cell signaling, so communication between cells, and they'll regulate the activities of other cells as well. So the three types that we'll look at are monokines, interleukins, and interferons. So starting off then, we're going to think about histamines. Histamines are released by the antigen-presenting cell, so they're represented here in red. They'll attract neutrophils at the site of infection, so they'll send a signal to the neutrophils, but they also cause leaky capillaries as well. Now, the antigen-presenting cell will also release another group of chemicals called monokines, which is in blue over here. So these are the monokines. Those will also attract neutrophils to the site of infection through a process that's called chemotaxis. So chemotaxis is when a cell is attracted to a particular chemical. Like if I put a pile of chocolate in the corner of the room and everyone starts to migrate towards that chocolate, that would be chemotaxis. Those monokines will also activate B cells and stimulate them to differentiate into B plasma cells, which will then produce antibodies. So next we'll look at the interleukins. So interleukins then, inter means between, and leukins refers to white blood cells, otherwise known as leukocytes. So that's why they're called interleukins. When that process of clonal selection takes place, the T helper cell will release interleukins and they'll send a message to the antigen-presenting cell. Antigen-presenting cell will send a message back then to the T helper cell. T helper cell then will release interleukins that will activate the correct type of B cell, so B cell that has got the same complementary receptors on its surface here, and the correct type of T cells, again, that have those complementary receptors on the surface. And those cells will send messages to each other. So that will activate those cells, and then they'll divide by mitosis and differentiate into the different cell types that we've got here. Finally then, we've got the interferons. So interferons have got a couple of jobs. They are released from the antigen-presenting cell and the infected cells as well, and they will inhibit viral replication. So that's one of the really important jobs that they have. But also, they will stimulate the T killer cells and encourage those T killer cells then to produce more of those toxins so that they're going to be destroying more of the pathogens and more of those infected cells. So we have got the monokines then that are released from the macrophages that will attract neutrophils through the process of chemotaxis when a chemical moves towards a particular cell moves towards a particular chemical. You've got monokines then that will stimulate the B cells to differentiate into B plasma cells and then release antibodies. Interleukins then released from B cells and T cells and macrophages, and they will stimulate the clonal expansion process. So cells will divide by mitosis and then they'll differentiate into the different cell types. And then interferons will stop viral replication by inhibiting that replication process, and they'll also stimulate the activity of T killer cells.